All right, guys, welcome back. So, we're going to wrap up our discussion of osmosis today, really just talking about the fact that when we're dealing with these different types of transport, we're talking about your cells being able to maintain homeostasis inside and outside of their cells. And so they're constantly at odds with their internal and external environments. And your cells, whether they're single celled organisms or multicellular celled organisms, have to have had ways that have evolved over time to deal with this water balance. And so, just a couple of quick examples of specialized methods that cells have developed over evolutionary history. So, if we look at something like a protist, these guys are single celled eukaryotic organisms. They live and are constantly surrounded by water. So, as they move around and do their day to day activity, they're going to find themselves in different environments. Sometimes it'll be isotonic, sometimes it'll be hypertonic, hypotonic, whatever the situation may be. Now, they're essentially animal cells. So, the only thing separating them from their environment is their cell membrane. And so, if they find themselves in a hypotonic environment, water's going to start rushing into their cells and they're going to start swelling up. And that's not necessarily a good thing. There's no way to keep them from over expanding. And it's essentially like filling up a water balloon. You can only put so much water into that balloon before eventually it's going to burst. There's nothing that's going to prevent that bursting from happening. So these protists, as a way to survive in those potentially hypotonic situations, they have actually evolved those contractile vacuoles that we talked about last unit. So remember that these contractile vacuoles are sort of like a sump pump in your basement. The pump sits in your basement. If water gets in, it turns that pump on. That pump's job is to evacuate or pump the water out of your basement. Well, for the protists, if they get into situations where they are in a hypotonic environment, their contractile vacuoles will contract more. So, the more water flowing into their cells, the more contractions occur by that vacuole, the more water gets expelled from the cell to hopefully help them maintain shape. So, if they're in a really hypertonic environment, meaning water is actually getting sucked out, the contraction rate is really low. They end up in an isotonic situation where water's sort of moving in at a constant rate. Those contractions will be at sort of a mid rate. And then if they end up in a hypotonic environment, those contractions will increase even more because now water is rushing in. All right, so kind of a good test question. If I take a look at the number of contractions on a protist, can I make a hypothesis as to what sort of environment that cell is finding itself in based on the number of contractions that are occurring? Your saltwater fish, go back to that clip I showed you earlier from Ice Age, right? You're constantly surrounded by water and you can't drink it. It's not going to be good to be next to your cells. Your cells need a proper balance. So these saltwater fish have figured out or have evolved these specialized gills that actually will pump the excess salt out so that they're getting the water crossing their gills so that they can do their oxygen exchange and breathe, but that the water that's actually passing along their gill cells themselves are at a much more isotonic salt to water ratio, so that they're not actually dehydrating those cells. And your plant cells also have an interesting situation because for them, remember, they've got this extra layer of their cell wall externally to their cell membrane. But what they don't have, unlike organisms like us, is a skeleton. So, in order for them to be able to maintain that upright stature as a plant, they have to have some other way to do it because they don't have bones. That's what our bones do for us. That skeletal structure inside our body connects to our muscles, provides us with posture and support, and that ability to maintain an upright position. Your plants don't have that. What they do have, however, is that cellulosic cell wall. And what that cell wall ends up doing for your individual plant cells is actually sort of act like a girdle. So, unlike those protists who could take in water forever until they actually explode, your plant cells can't do that. Their cell wall is going to prevent that from happening. So, what's going to happen is when they get into a hypotonic environment and that water is rushing into their cells, we're going to fill up that central vacuole. That central vacuole is going to swell up. It's going to cause excess pressure to build and push out against the outside of that cell. But unlike that protist, who that pressure would eventually get too great and just burst, your plant cells actually will build up that pressure, push out against that cell wall, but that cell wall will push back. All right. And so what ends up happening is we build up this pressure in your plant cells called turgor pressure. And it's actually a good thing for them. All right, a proper level of turgor pressure, pressure from water in their central vacuole pushing out against that cell wall, actually provides the cell that structure and rigidity like the bones do for our body. And so if that central vacuole is nice and full, that turgor pressure is nice and high, all right, that's going to allow your plants to stand up. So that's what you're seeing down here, right? So turgid cell, really full central vacuole pushing out against that cell wall, and your plants can stand up. Now, if you forget to water your plants for a few days and they start losing that water and that central vacuole starts to shrink, all right, which is what's happening here, that pressure decreases because that cell wall is now going to exert more pressure towards the center of the cell. And what happens to your plants? They fall over, right? They wilt. What we would talk about as a flaccid cell instead. So if we combined all of this information in terms of what environment's best for different cells, there are differences. All right. Not every cell is necessarily going to be ideally in one situation. So if we just narrow it down to a plant versus an animal cell and compare what would be the best situation versus that isotonic, hypertonic, hypotonic, that's sort of what this chart is trying to do for you. So if I take a look at an isotonic solution, right? Remember the amount of water and solute on either side of the membrane is identical. So water is moving in and out at the same rate. So for a plant cell, it's okay. It's not the best situation. All right, their cells would be what we would label as flaccid. All right, they're sort of limp, wilting. They're not necessarily dead, but they don't really have that rigidity to be able to stand up. So not really the ideal situation for a plant cell. For an animal cell, however, something like us, we need to be in that isotonic situation. We need the same amount of water moving in and out of our cells at a constant rate so that the cells can maintain shape. That's a good thing for them. All right. Go down to hypertonic. Right now, there's lots and lots of solute outside of my cells, not a whole lot of water. So the water is moving from inside my cells to out. The cells are shrinking. And this is a bad situation for either type of cell. They're being dehydrated, they're losing water. That's not a good thing. Your cells need a certain amount of water to maintain their daily function. So if I see that situation in a plant cell, we can talk about it strictly as just being dehydrated, but if it dehydrates enough where the cell membrane actually separates from the cell wall, you'll come across this term called plasmolysing. All right, where the plasma membrane lyses or breaks away from that cell wall. And that's not a good thing. Once your plants get to that situation, there's really no way to come back from it. And your cells are going to end up dying. Your plant's going to end up dying as a result. An animal cell, same thing. Not a good situation for us either. So they will also end up dehydrating, or you'll come across this term called crenated. Often you'll see it in diagrams with red blood cells where these cells look all shriveled and crinkled up. Once again, not a good situation. That's not where we want to be. Go down to hypotonic, and now remember that this means that there's not a lot of solute in their environment, but a lot of water instead. So that water ends up rushing into your cells swell up. All right. This is the best situation for your plant cells. This is where your cells are able to build up that turgor pressure, push out against that cell wall, and it gives that whole plant its stability, rigidity, and be able to stand. Animal cells, on the other hand, bad idea. This is like the overfilled water balloon. If I keep pumping water into my animal cells, they don't have that extra layer, that extra support of a cell wall, and so eventually they end up bursting, all right, or what we call lysing the cell. Not what we want to do, not a good situation. So isotonic is best for animal cells, but the hypotonic is actually best for your plant cells, and hypertonic is bad for everyone.